Hi there everyone. In today's video we are going to talk about the special senses. We are going to study the physiology of smell under this topic. So we're going to see the definition, the receptors, the olfactory pathways, the factors affecting the olfaction and the reflexes, and then the applied aspect. So this is going to be very easy, simple to understand, containing the flowcharts and all the diagrams. So let us begin. The sense of smell is known as olfaction. Okay, the smell that we sense, it is called as the olfaction. Okay, it is called as olfaction. Now it is classified as visceral function as it is closely related to the gastrointestinal function. Okay, it is related to the gastrointestinal function. That is the reason it is classified as the visceral function. Now, the receptors, talking about the receptors, they are called as the olfactory receptors. The receptors for the olfaction are the chemoreceptors, okay, by the chemoreceptors through the release of neurotransmitters that can be inhibitory or excitatory. Fine. Now, they are located in the olfactory mucosa of the nasal epithelium. Okay, our nose has epithelium and it also has the mucosa. That mucosa is called as the olfactory mucosa. So, these chemoreceptors, they are located in the olfactory mucosa of the nasal epithelium. Now, talking about the animals, animals have a well-developed sensations of the smell. Okay, the sense of smell is well defined in animals. This helps them to locate their mates for the process of mating, differencing between the friends and the foes. So we take an example of a cat and a mouse. So the cat chases the mouse, right? The cat chases the mouse based on the sense of smell, sensation and perception of the smell. I don't know, I'm stammering today. So identification of the territory, like where they belong to and stuff. So these are the reasons the animals have well developed sensation. In humans, we can sense the smell of food, perfumes, deodorants, flowers, etc. And it is helpful for enhancing social and emotional interaction. Okay, social and emotional interaction with the people and animals as well. Enrichment of the memory. Okay, enrichment of the memory. Now if you just think about eating a cake, okay, if I tell you to think of a chocolate cake, you will kind of sense the taste and also kind of sense the smell of the chocolate cake, even though the chocolate cake is not present in front of you. And if I were to think of a strawberry cake, you would surely sense the smell of the strawberry even though the strawberry is not present. So it is because of the memory. Okay, it enriches the memory as well. And staying away from the stale food, the food that is not good, okay, the food that contains harmful chemicals, the food that is not good to smell, not good to taste. Staying away from the food and polluted environments. Polluted environments, if you go to more disturbed area, like there is a lot of smoke, there is a lot of pollution, air pollution and stuff. So it irritates your nose, it irritates the nasal cavity. So to stay away from the polluted area, it is a body's mechanism to tell you that this area is polluted and you should shift to other area where the air is fresh and your nasal cavity is not getting irritated. Okay, now the olfactory sensory system is the only system that does not rely on thalamus. Okay, so we will see this point under the section of pathways, the olfactory pathways. Okay, olfactory pathways. It has asymmetrical cortical representation. We will see what is asymmetrical, why is it asymmetrical, what is the pathway, how does it get transported, transduction, olfactory transduction. We'll see all that. Now the site of olfaction, the olfactory receptor cells are present in the mucosa of the olfactory mucosa of the nasal epithelium, as we already mentioned. So its location is, it is a yellowish pigmented mucous membrane. Okay, yellowish pigmented mucous membrane that is present in the nasal epithelium. So it is extending medially, it is extending in two ways, two sides. That is, at one side it is laterally extending, and at one side it is medial extending. So the lateral it is extending to the superior concha, and the medially it is extending to the upper one third of the median septum. Okay, the septum is nothing but the area or the region or the skin that separates the two nostrils. Okay, so upper one third of the median septum. Okay, they are extending. This is a pigmented mucus that is extending laterally to the superior concha and medially to the upper one third of the median septum. Now it is covered by the Bowman's glands. Okay, there is specialized glands we have, those are called as the Bowman's gland. Please mark them, they are important. Okay, this can be asked in MCQ as well. It covers 2.5 centimeters square in each nostril, so of course 2 centimeters square in both the nostrils. Okay, so what are macrosomatic animal? Who are the macrosomatic animals? The area is covered more in the animals. Okay, suppose we have 2.5, they might be having 3, they might be having 5, they might be having 7, so they are called the macrosomatic animals. Okay, higher sense and perception of smell, example dogs. Okay, the animals which have higher perception of smell, and the area covered by the mucus is more, or the Bowman's gland is more, they are the macrosomatic animals. For example, it is the dogs. And a important point to be noted, that humans are microsomatic. Okay, the animals like dogs, cats are the macrosomatic, and humans are microsomatic, because they cover the mucus covers a lesser area, that is only 2.5 centimeters square in each nostril. Okay, now the olfactory epithelium contains around 10 to 20 million receptor cells. Okay, 10 to 20 millions. Okay, remember this, 10 to 20 million receptor cells, and by this we can sense around 10,000 different types of odors. Okay, 10,000 different types of odor, we will see that under this section. Okay, few are the progenitor cells. Among this 10 to 20 million receptor cells, we have few progenitor cells which give rise to new receptor cells by the process of mitosis. Okay, the doubling, by the process of mitosis. Fine. Now an important point to be noted, that sniffing is a semi-reflex response. Okay, sniffing is a semi-reflex response that is evoked to noble odors. Okay, suppose, okay, again, I love cakes and chocolates. Okay, especially a chocolate cake. So if my mama would bring me a cake, okay, then just by the perception of the smell, the perception of the odor that the cake is producing, that is the noble odor, that will evoke a response, that is sniffing. And sniffing is a semi-reflex response. Okay, that is going, the odor, the pleasant odor, it is going to awake the sniffing, which is a semi-reflex response. Okay, this is important to know. And what are the vomeronasal organs? Okay, they are the extra layer of olfactory nasal mucous membrane, rodents, present in the rodents and the other mammals. Okay, they are the extra layer of olfactory nasal mucous membrane. So there's nothing but an extra nasal mucous membrane, that is an extra layer that is present in the nasal epithelium again. So that is only called as the vomeronasal organ, and it helps in identification of the pheromones. Pheromones are nothing but they are the hormones. Okay, they are hormone like substances related to olfaction. Okay, responsible for mating in the animals. Okay, it is in the animals, the in mammals it is present in the mammals as well as in the rodents as well. So it helps in identification of the pheromones of the same species. Okay, not the different species, responsible for mating. It is specific in species. Okay, so now the rodents and rodent species will be having specific type of pheromones, and the humans will be having specific type of pheromones, and they're directly related to, pheromones are directly from your DNA. Okay, from the DNA, and they are hormone like substances. Now axons of the vomeronasal organ go to the accessory olfactory bulb. We will study, okay, the what is the olfactory bulb, olfactory epithelium, olfactory glomeruli, and different things. Okay, then it goes to the hypothalamus and the amygdala, and it helps in it evokes the response, it evokes, or it increases the, or it stimulates the reproductive and the feeding behavior which one? The vomeronasal gland through the accessory olfactory bulb to the hypothalamus and amygdala, which evokes or stimulates the reproductive and the feeding behavior. Okay, that is why it is told that it helps in mating. It helps in mating in who? The rodents and the other mammals, which that is the vomeronasal organs. Okay, now talking about the location of the olfactory mucous membrane. So see, suppose this is your nose. Okay, wait, this is your nose. Let me take a darker shade. Okay, so this is your nose and this is your upper lip. Okay, this is a superior and medial inferior tubernates. This is a nasopharynx, whatever we see here, and this is the hard palate. Okay, this is the hard palate, and see this one yellowish color, whatever we studied just now, that is the 2.5 centimeter squaring in each nostril. Okay, this yellowish pigmented mucus is nothing but the olfactory mucous membrane, and the double layer, the extra layer is present in the rodents and the mammals, which is called as the vomeronasal organ that we just now studied. Now, so this one is the olfactory bulb, and these are the nerves, that is the olfactory nerve, and the tract, the olfactory tract is nothing but from the axons to the the cortex, whatever pathway is there it is carrying, that is only called as the olfactory tract. Okay, that is the olfactory tract. We have olfactory bulb, olfactory nerve, olfactory tract, and we have the olfactory mucous membrane. Okay, so this is the location of the olfactory mucous membrane. So just draw this diagram in the exam when they ask you to when they ask about the location of the olfactory mucous membrane. Now talking, this is my book, okay, and this is my dirty handwriting that I've written in the corner. Okay, olfactory receptor cells and the olfactory pathway. So what is the pathway? So one important thing that we have to know, this is the cribriform plate. Okay, we know the ethmoid bone is there, and near that we have the cribriform plate. Cribriform plate is important. Okay, so these are the cilia. We have around 10 to 20 cilia on each receptor cell. Okay, 10 to 20 cilia on each receptor cell, which help in carrying the odorant. Okay, so these are the receptor cells and these are the cilia, 10 to 20 cilia, and these are the supporting cells and these are the mitochondria for the production of energy. And then we have the olfactory nerve fibers. Okay, the axons and the receptor cells bind to form 20 bundles. Okay, 20 bundles of nerve fibers. We will study that. Okay, and this is the olfactory bulb, which is actually looking like a bulb. So this is the olfactory bulb, and what do you see here? These things are the olfactory glomerulus. Okay, these have major role in inhibition and excitation. We'll study that. And this is the ethmoid bone, and this is the cribriform plate in which the olfactory nerve fibers are piercing. Okay, this goes to the olfactory cortex. Okay, this goes to the olfactory cortex. So from the cilia to the receptor cell to the olfactory fibers, from the olfactory fiber to the cribriform plate, from the cribriform plate to the olfactory glomeruli, from there to the olfactory bulb, and directly to the olfactory cortex. Okay, directly to the olfactory cortex. Let me erase that so that it will be easy for you to see. Perfect. And let me also take this off. Perfect. So now let us study about the olfactory receptor cells. They are bipolar. Let's do it fast because it is very easy topic. Bipolar, short and thick dendrite. They have short and thick dendrite, and the synapse that is formed is of the type axodendritic synapse. Okay, axon, presynaptic axon and postsynaptic dendrite. Okay, expanded to form the olfactory rod, which gives off 10 to 20 cilia, as I already mentioned. Okay, so suppose this is the axon, receptor cell is also present, so it has 10 to 20 cilia. Okay, 10 to 20 cilia, and they are expanded, which we call the olfactory rod, each of which gives 10 to 20 cilia. Now these olfactory and taste related neurons are unique. They are unique. Okay, they are unique because they have a property of regeneration. We saw about the progenitor cell, the basal proliferation cells. Each of them is replaced every 60 days. Each of them is replaced every 60 days, that is approximately 2 months, due to the basal cell proliferation that I already mentioned, which forms the new receptors. Okay, now what is BMP? Okay, that is BMP, bone morphogenetic protein. Okay, what does the bone morphogenetic protein do? Here the nervous system is exposed to the external environment. Okay, the nervous system which is exposed to the external environment is called the bone, due to the bone morphogenetic protein. Now talking about the olfactory pathways. Okay, this is very important to know. If you don't understand this, then the whole chapter will be worthless to study. Okay, so olfactory pathways, these are very important. So talking about the olfactory nerves, they are the receptors gather together. As I already mentioned, the receptor gathers to form 20 bundle of nerves by piercing the cribriform plate and the ethmoid bone to form the olfactory bulb. See, I already showed you in the diagram, right? So see, this is the cribriform plate. From the cribriform plate, it pierces into the ethmoid bone. These are the olfactory. This is the cilia. This is receptor cells. Olfactory fibers pierce the cribriform plate of the ethmoid bone. Then go to the olfactory glomerulus and reach the olfactory bulb. So this is what it is written in words. See, receptor gather to form 20 bundle of nerves by piercing the cribriform plate and the ethmoid bone. They reach the olfactory bulb. So this is about the nerves. Olfactory bulb. Okay, what is olfactory bulb? It is present above cribriform plate and below the orbital surface of the frontal lobe on each side. Okay, orbital surface you have studied in anatomy, and frontal lobe on each side is present. Okay, so what it does, it has 4 types of cell. 4 very important types of cells, that is the small tufted cells and large mitral cells. You can only remember tufted cells and mitral cells. Okay, they are the second order neurons. Okay, they are the second order neurons. Which one? The tufted cells and the mitral cells. Okay, and the periglomerular cells and the granule cells, they are the interneurons which help in inhibition. Okay, inhibition. So these are the interneurons and these are the second order neurons. Okay, so these are the second order neurons and these are the interneurons. Now we are going to talk about the olfactory glomeruli. They are the exodendritic dendritic synapses I already mentioned, and it can do the excitation and can also do the inhibition. Okay, excitation by the first order neuron. When the first order neuron converges onto the second order neuron, excitation happens, and if there is high convergence, of the first order neuron onto the second order neuron, it leads to increased sensitivity. Okay, increased sensitivity, it leads to increased sensitivity. This is about the excitation. And for the inhibition, the mitral, this is a very important flow chart to understand, because in GK PAL it is slightly given in a confused way. So I have broken it down into a flow chart. So in the mitral and tufted cells stimulate the granule cells. See, mitral and tufted cells, they are the second order neurons, and the granule cells, they are the interneurons. The mitral and tufted cells, they stimulate the granule cells by the neurotransmitter, that is glutamate. In neurophysiology we studied that glutamate is a excitatory neurotransmitter. Okay, and in turn the granule cells, they inhibit the mitral and tufted cells, okay, by the GABA, that is the gamma aminobutyric acid, which is a type of inhibitory neurotransmitter, again studied in the neurophysiology. So this is how you need to relate the things. So the mitral and tufted cells, they stimulate the granule cells through glutamate, they excite it, and in turn the granule cell, they inhibit the mitral and tufted cells by the inhibitory neurotransmitter, that is the gamma aminobutyric acid. Important to remember. Okay, so this is about the olfactory glomeruli, what it does, what are the functions and what it is. And talking about the olfactory tract, the axons are extended to olfactory cortical areas. This is only called as the olfactory tract. The receptors and the axons from here to the olfactory cortical areas, they are the olfactory tract. Okay, olfactory tract, fine. Now talking about the cortical olfactory areas, while entering brain, it divides into the lateral olfactory striae and the medial olfactory striae. Which one? The cortical olfactory areas. Okay, the lateral and the medial, let us understand now with this. Okay, the lateral and medial. Lateral we have remember 5. 1 2 3 4. Okay, 4 and one more, you remember 5. Okay, we have the piriformis cortex, amygdala, and we have the entorhinal cortex, lateral and anterior orbitofrontal cortex. And for the medial olfactory striae, we have the it generates autonomic response, that is mainly the parasympathetic. Olfactory fiber project to the dorsal motor nuclei of the vagus. Okay, dorsal motor nucleus of the vagus stimulates the parasympathetic response, which helps in secretion of saliva, gastric and pancreatic juices whenever we smell something pleasant or some foods that we really like. Okay, so this is the parasympathetic response by the stimulation of the dorsal motor nucleus of vagus. Okay, so this is due to the medial olfactory striae. And for the lateral olfactory, we have piriformis cortex that helps in sniffing, amygdala in the emotional response, entorhinal cortex for memories of the smell and stuff, which I already mentioned. Lateral and anterior orbitofrontal cortex helps in perception and differentiation of the odors. Okay, differentiation of the odors. Now let us understand by the flowchart. So see, tufted cells and the mitral cells. Tufted cells and the mitral cells are from the olfactory bulb. These are the second order neurons. They travel through the lateral olfactory striae, go to the piriformis cortex, entorhinal cortex, amygdala, olfactory tubercle and the anterior olfactory nucleus. These you need and must and should remember all this. Okay, so from here these tufted cells can directly also go to the olfactory tubercle as well. This is about the lateral olfactory striae. From the medial, it comes from the mitral cells. These are the accessory olfactory bulb has a mitral cells also. From here, from the medial olfactory striae, it goes to the amygdala, only the amygdala, which can stimulate the hypothalamus and directly stimulate the frontal cortex. We have two cortexes, orbitofrontal cortex and the frontal cortex. Okay, the hippocampus from the entorhinal and the hypothalamus from the amygdala can stimulate the frontal cortex, and it can directly stimulate as well. And see, from the amygdala, from the entorhinal cortex, from the piriformis cortex, from the olfactory tubercle, it is sending to the thalamus. It is going to the thalamus and finally to the orbitofrontal cortex. But we have to note one very important thing, that the anterior olfactory nucleus doesn't pass to the thalamus, while all the other do, but this does not. Okay, so see, it goes to from the olfactory bulb, from the second order neuron, lateral olfactory striae goes to the amygdala, entorhinal cortex, piriformis cortex, olfactory tubercle and the anterior olfactory nucleus. From tufted cell, it can go to olfactory tubercle and anterior olfactory nucleus as well. Then it goes to the thalamus. From all these four, it goes to the thalamus and the finally orbitofrontal cortex. And from the amygdala, it goes to the hypothalamus and it can directly go to the frontal cortex. And from the entorhinal cortex, it can go to the hippocampus. Okay, from the to the hippocampus. And the only nucleus that doesn't go or send to the thalamus is the anterior olfactory nucleus. Very important to remember. In turn, it helps in it goes to the contralateral olfactory bulb. So this is the pathway. So done with the pathway. Now physiology of olfaction. What is the physiology of olfaction? We have two in this: role of the mucus odorant binding protein and the olfactory transduction. Okay, let us understand fastly. Receptors are activated when odoriferous substances are dissolved in thin layers of mucus covering olfactory epithelium and come in contact with the cilia. So we all know that our nasal is there. Suppose this is a nose, and there is presence of yellowish pigmented mucus, and this mucus, whenever the odoriferous substance that is volatile and is in the liquid form, it dissolves and it gets volatile, and then it reaches the mucus covering of the nose. It gets dissolved in the olfactory epithelium and it comes in contact of the cilia, and this is how perception of the smell begins. Now the odoriferous substance are small molecule with carbon atoms. Okay, the lipid soluble odorant bind to the odorant binding protein, that is OBP. Odorant binding protein that is present, OBP, is secreted into the mucus by the lateral nasal gland. So this is about the role of the mucus and odorant binding protein, that is OBP. Talk about the olfactory transduction. Okay, so there are 1000 different odorant receptors that are coupled to heterometric G protein. Okay, there is a thing called G protein that we have studied in the endocrine physiology. 1000 different odorant receptors are coupled to heterometric G protein. This G protein get activated when they get coupled, and opening of the cation channel takes place. After opening of the cation channels, influx of the calcium takes place. This is the major mechanism. Okay, this mechanism is the major mechanism, and the adenyl cyclase, that is CMP system, gets activated by the G protein, which is by we write it in the subscript. Okay, OLF. Okay, now this stimulates the phospholipase. Okay, this stimulates the phospholipase and it causes depolarization. Action potential is generated. So this is the mechanism of the olfactory transduction. What happens? They are coupled to the heterometric G protein. The G protein gets activated, the calcium channels, the cation channels get opened, influx of the calcium ion takes place, which is a major step or the major mechanism. Adenyl cyclase system gets activated. This in turn stimulates the phospholipase and depolarization occurs and the action potential is generated. So this is how the olfactory transduction takes place. Now, talking about the sharpening of the impulses, there's a term that is called as the sharpening of impulses. What happens is there is continuous background firing of impulses through the olfactory tract. Okay, to the olfactory tract, which increases the frequency. The frequency increases, the periglomerular granule cells, they spread laterally. Okay, the periglomerular cells, they spread laterally, which causes inhibition of the olfactory glomuli and the sharpening of the action potential in tufted cells and the mitral cells, which are the second order neurons. Okay, the excitation of this takes place, whereas inhibition of the olfactory glomeruli happens. This is due to the increased frequency of the continuous background firing. This is only called as the sharpening of impulses. Now, talking about the discrimination of odors, there are two factors responsible, very important. The two factors are various type of odorant receptors are present, that is around 10,000 receptors are present for different sensing and perception of different types of smells, and different olfactory neurons project into different glomeruli. So one neuron, it projects into one glomeruli, and the second one into the another glomeruli. So different olfactory neurons project into different glomeruli. So this is the reason that discrimination of the odors is possible. Now we are going to talk about the factors that are affecting the olfaction. So there are 4 factors. This is 1, 2, 3, 4 factors are there. Okay, there are 4 factors. Now, type of odorant stimuli, easily transported to the nose via the air, which is quickly recognized. Okay, if it is volatile, the odorant it is transported to the nose via the air, which is quickly recognized. Strong odor has high lipid and water solubility, high lipid or the water solubility. So this is about the type of odorant stimulus. Okay, talking about the threshold of the olfactory receptors, extremely sensitive to minute concentration of odorant as well. So for example, we have this methyl mercaptan can be sensed with the concentration of only 0.000004 milligram per liter of air. So this is such a less concentration, such very less concentration. Still we can still the perception of smell is possible due to the threshold of the olfactory receptors. Now, women have higher sensitivity of the perception and differentiation of the smell than males, especially during the process of ovulation, and threshold increases with age in both females and males. So, an important point to note, this is a box that you have to remember. It will be very easy to understand. And remember, threshold is directly proportional to age. As the age rises, the threshold also rises. The sensation or the perception decreases with age. Okay, as the age increases, the sensation and perception decreases. So this is the inverse proportional, and this is the direct proportional. Okay, so this is the second factor. The third factor is intensity of the stimulation or stimulant. We can discriminate over 1000 different odors, but the ability to sense the change in intensity, only single. Suppose you take clove oil and you are told to sense the intensity of the odor it is insignificant. Okay, it is insignificant, you can't do that. Change in the concentration of odorant must be over 30% to perceive the change in intensity. So this is about the intensity of stimulus. Now talking about the last one, that is the olfactory adaptation. As the name suggests, when the olfactory receptors are continuously stimulated, they adapt rapidly. Okay, they adapt rapidly when they are continuously stimulated due to cyclic nucleotide gated ions. Okay, due to this. Okay, the cyclic nucleotide gated CNG ions by the calcium calmodulin impulses. So when this calcium complexes are reduced, not impulse complex. Okay, when it is reduced, it makes adaptation slower. Okay, when it is reduced, it makes the adaptation slower. So these are the 4 factors affecting the olfaction. Now moving to the next one faster, we have the reflex associated with the olfaction. Sniffing at arrival of the odorant, especially when sniffing, as we already studied, it is a semi-reflex response. It is a semi-reflex response to what? To pleasant odor. Okay, pleasant odor or smell of your delicious food, your favorite food. Okay, the second one is at smell of food, salivation, gastric and pancreatic secretion increases. Okay, when you think about ice cream, or you think about your cakes, or think about your favorite biryani, so the salivation, your mouth gets filled with saliva. That is due to the smell. Okay, the smell of the food. This is the reflex associated with olfaction. And vomiting in response to foul smell. Okay, the smell is very dirty, it's very unpleasant, and it is very strong, so you can't even bear it, so that leads to vomiting. Okay, that is also a reflex response. Now sneezing and lacrimation. Okay, sneezing irritates the cilia, okay, that are present in the nasal epithelium, and lacrimation falling of the tears, and in severe cases respiratory inhibition, because it is a protective mechanism of our body, that our body is compensating, okay, in response to irritant odorants. Okay, the odors that are very irritant, that are very bad for your health, like we have this chlorine. As we have this chlorine, we have ammonia, a very unpleasant smell, methanol, and the peppermint. Okay, although peppermint has a mild, not bad odor. It stimulates the naked nerve endings of the trigeminal pain fiber. This is the 5th cranial nerve. It gets stimulated. Unpleasant or burning or vomiting sensations are felt because the 5th cranial nerve is stimulated due to the irritant odors. Okay, and also sneezing and lacrimation is also seen. Now applied aspect, we have mainly 4, that is a hyposmia, anosmia, dysmosmia and pyrosmia. Okay, let us understand. Hyposmia is nothing but diminished sense. Hypo, less, or diminished sense of smell. Anosmia, absent, a means absent, so absence of the smell. Okay, it depends upon the structural or functional loss of sense of smell. It depends, okay, if it is structural, if it functions, or if intensity of the loss. So if it is more, then it causes anosmia, and if it is less, it causes hyposmia. Okay, now talking about the dysmosmia and parosmia. So dysosmia is distorted smell. The person cannot differentiate properly between two smells. Now if you give 2 odors, that is clove oil and the peppermint oil, he or she cannot differentiate properly. Distorted, it mixed sensations of the smells. Now talking about the parosmia, we have perverted smell. Okay, offensive odors feel pleasant and vice versa. The offensive odors which are unpleasant might feel pleasant for some people, and the pleasant one feel unpleasant for this few people. So this is called as the parosmia. This is called as parosmia. It is due to the temporal lobe epilepsy, seizures, lesions and all. The temporal lobe is present. Okay, temporal lobe epilepsy also causes olfactory hallucination. Hallucination is imagining or seeing something, or listening, or smelling something that actually doesn't exist. Okay, it is due to the temporal lobe epilepsy. Imaginary repulsive smell with involuntary movements of lips and tongue. Okay, this is involved. So this is about the applied aspects of the hyposmia, anosmia, dysmosmia and parosmia. And see, the anosmia and the hyponosmia can be caused by common cold. Okay, when there is common cold and all, you won't perceive this smell. Okay, you won't get it. The sensations will decrease a little. It is the most commonest cause of hyposmia. Old age, fracture or injury to the cribriform plate that we have seen, that is present in the ethmoid bone, or these all other olfactory pathways like glomeruli, the olfactory bulb, the nucleuses. Injury to these things, these structures can also lead to dysosmia or directly anosmia. Okay, when there is combined anosmia, that is complete sensational loss of the smell, and hypogonadism, it causes this syndrome is named as the Kalman syndrome. Okay, the Kalman syndrome, the anosmia and the hypogonadism, when they both occur at once, so there is a particular syndrome that is called as the Kalman syndrome. Okay, now test for olfaction. This is the last topic. Person's eye must be closed. This we have done in the practicals. Of the cranial nerve. So test for olfaction. Person must close his or her eyes. Small bottles containing the peppermint and clove oil are used, and they are sealed with the rubber airtight caps or airtight covers, so that it doesn't it is volatile. So the odorant, whatever it is there, it should be dissolved. Okay, it should be in liquid form, and it should be volatile. These are the two conditions to be fulfilled. A peppermint and the clove oil. The person must close his or her eyes. Small bottles containing the peppermint and the clove oil are used, which are covered and tightly closed by the rubber airtight caps, and when you remove it, must be presented to each nostril separately. Okay, you must take the small bottle to the person's nose, okay, the nostril, so that he or she could smell it. Now this leads to the qualitative test for olfaction, and then you have to ask the person which oil was it clove oil or was it peppermint oil. If he or she answers it correctly, their nerves are working well, but if not, it is distorted, they are unable to differentiate between the two, or they are mixing it, or they are reversing. Like if they are calling the peppermint oil clove oil, and to the clove oil they are calling peppermint oil, they can be a victim of the dysosmia. Okay, now, and if odorant is irritant, the fifth cranial nerve is stimulated, as I already told. So these are non-irritating. The peppermint and the clove oil, they are not irritating as such when dissolved. Okay, qualitative test for the olfaction. So if the odorants are irritative, they lead to activation of the stimulus to the fifth cranial nerve. So when the fifth cranial nerve gets stimulated, it leads to sneezing, it leads to lacrimination, tears falling off. It can also lead to vomiting and burning sensations as well. Now the quantitative test is done using the olfactometer. Which meter? The olfactometer, because it's olfactory. So the olfactometer. The qualitative is done by the olfaction, by telling the person to close his or her eyes, and giving them a chance to smell the oils or smell the odorant. So this is about the chapter. Thank you so much for watching, and let me know in the comments which video shall I make next.